genomics is not this elusive field that we just study in our PhDs theoretically and that's it. It is really being used in everyday life and is impacting people like you and me. So very simply put, all of the 3 billion cells in our body, except the red blood cells, have a nucleus. Inside that nucleus, you have chromosomes. And those chromosomes are nothing but structures formed by your DNA, right? And your entire set of DNA that is replicated in every single cell in your body is called the genome. When we are studying this entire set of DNA of any person, that branch of study is called genomics, which is what I do. Within the DNA, you can think of how does the DNA really determine that I have brown eyes and somebody else has blue eyes, or I'm short and the other person's tall, or any other noticeable or unnoticeable trait about me. It's because the DNA is organized into functional units called the gene. Some people focus their efforts only on understanding that these genes, when they're passed down from your mom and dad, how are they impacting you? So that study of inheritance is called genetics. But if we focus on only you and what you have in terms of your DNA makeup and the genes, and we try and help understand that, if we alter it, then how does it impact your health? How does it impact your physical traits? Then that's just called genomics. So back in 2003, actually back in 1991, a bunch of researchers decided that we keep studying genetics, in terms of diseases or physical traits, but we really don't know that if we take one person and try and study the genome, what does it look like? We have some understanding through our experiments, but not completely. So what they did is they launched like a really decade-long experiment, and they asked for blood from multiple volunteers. So you can imagine there were like a bunch of hospitals in the US, and they decided, okay, we're going to do this. And they just asked for volunteers around the hospital to donate blood. So and it was all anonymized. We don't really know who donated, whose blood was finally used. But people came in, they donated their blood, and using those samples, nearly 2,800 scientists around the world tried to sequence the human genome for the first time. Till today, it's not complete. We managed to sequence it for up to 99%. But that first map of what the DNA is in any human being was published in 2003. So when I say the human genome was sequenced, I mean, if I take myself and I take out a blood sample, and within that blood I have my DNA, I can know exactly how that DNA reads. So I don't know if you guys are aware or not, but the DNA is made up of 4 basic molecules, A, T, C, G. And you can think of it as like a really, really long textbook. If you just unwrap the DNA into this really long string, it would just keep reading ATCG, ATCG in some random but very meaningful manner. And it would be so long, you'd probably stretch it to the moon once. So they could actually know for any human being what that DNA sequence is. At the time, it took 13 years and $3 billion because it was painstakingly slow. We were learning how to do it. We were trying to understand, okay, which gene is on which chromosome, how are the chromosomes arranged, what does the sequence look like, what do these genes really mean? And you can imagine technology back then wasn't as great as today. But today, if I want to sequence your or mine genome, it takes a few hours. I'll take a blood sample from you, put it through a few processing steps, and under $1,000, that's about 70,000 rupees, I can plug a machine to my computer, just like you plug your hard disk or some gaming console, and get your sequence read out. And that's how it's sort of revolutionized the whole landscape of healthcare. It no longer takes 1 year or 13 years to do that. That's how we've progressed to where we are with the whole state of genomics today. But that doesn't mean that we are only studying our DNA. If you guys are aware, from the DNA, RNA is transcribed, which kind of is an instruction of what that gene is going to do. And then from the RNA, proteins are produced, which actually go and do stuff in your body, right? So when you see your blood is red, that's the hemoglobin protein. I don't know, you might have heard of CRP, or you might have heard of uric acid, stuff like that, right? So those are all proteins in the body, effectively going and doing work in the body. So we can now, using these machines, actually study the DNA sequence. We can study the RNA sequence. We can study what the proteins look like. We can even study more downstream products of the protein called metabolomics. And all of them together, we can integrate this information to understand why are you like you are. In a normal human being, it's only satisfying our curiosity, but in a diseased patient, it can help us understand what is going wrong in the body and how do we correct it. So I'll first pause here to see if you guys have any questions. No questions? Everything's super clear that I spoke about?
So Aditya has asked a question, Aditi, what does the genome sequence tell us?
Well, that's a very good question. How about I was just going to go there, but why don't we take a shot at you telling me what do you think, if you knew the DNA sequence, um, what do you think you'd do with it? I'm sorry, I can't see the chat here, so I don't
No,
know if
don't
there
worry.
are other questions.
Aditya, you can unmute yourself also. Not a problem. I guess structure of the body, that's what he's saying.
Structure of the body, yeah, okay. So let's go back to this slide, yeah, and then I'll give you some real life examples. um, what is in your DNA sequence is going to determine what genes are expressed in your body, which will determine what proteins are being formed in your body. That will determine how your cells are formed, um, what is the color of your skin, how healthy you are. Are you a fast runner? Are you a slow runner? Um, are you healthy? Are you not healthy? Right? So I'll come to examples, but everything that makes you is encoded in that DNA sequence. It's like a blueprint for the body. So, like you said, what is it? Can we really use it for? So let me give you an example of how it's used in cancer. The photo that you see here is of a researcher called Dr. Lucas Watman. And back in 2003, he was diagnosed with cancer. He felt really ill. He had a type of blood cancer where a lot of white blood cells were being produced in his body. In 2003, if you went to the doctor and he diagnosed you with blood cancer, they will give you chemotherapy, right? And those are really strong drugs that try and just brute force and kill these cancer cells in your body, but they also make you really, really unwell at the same time. He went through multiple rounds of that therapy, that chemotherapy, but the cancer kept coming back and he just kept falling ill. So obviously, doing just chemotherapy was not helping. But at that time, nobody really knew that what is it about, what really happens in leukemia, that these white blood cells are suddenly being produced 10 times more than they should be. But 2003 was also the year when Human Genome Project was just finished. There was a lot of knowledge about, how to sequence, how to get the DNA blueprint out. And so what they decided to do is they're like, okay, let's sequence these cells and see if the DNA can tell us something. So what they did is they took leukemia cells, those tumor cells from his body, and they also took some normal skin cells. Again, from an area that did not have the tumor, right? And they put it through these sequencing machines. And they sequence two things. They sequence the DNA, and then they sequence the downstream RNA. And when they sequenced both of them, now one thing is they just sequenced it and they're like, okay, what does it really mean, right? But remember, in 2003, they now have this human genome map. They know if we take the DNA of a healthy individual, what does it really look like? So they tried to compare the DNA sequence of Dr. Watman to those healthy individuals. When they looked at the DNA, everything kind of looked the same. There really wasn't anything that stood out. But when they looked at the RNA and tried to compare it, they found that there was a gene called FLT3, and it was overexpressed. There were a lot of copies of this gene in the body compared to what a normal healthy person would have. And then they already knew at the time that FLT3 is exactly what is used to generate white blood cells. So they made the correlation, okay, maybe because of this gene that's being overexpressed and producing so many white blood cells is what is driving Dr. Watman's cancer. There was no good drug for leukemia at the time, but there was a drug that was used to treat kidney cancer that essentially, in principle, what it did was it tried to block the expression of this gene. And when they gave, under the hypothesis that, if we give Dr. Watman this drug, effectively it will block the expression of this gene and stop his leukemia, that's exactly what happened. In 2 weeks of taking this drug, which was actually for kidney cancer, nowhere related to blood cancer, it started curing Dr. Watman. And he could actually see, like, his reports showed that leukemia was going down, the number of cancer cells was reducing, and he actually got better and healthier. So that's one real life example of what can be learned if we look at the DNA.
I have a question here, Aditi. So how do you know what part of that sequence corresponds to what part of the body? Like, because it's only four proteins. I'm talking DNA sequence. So
Mm-hmm.
how do you know if this is to do with my eye, expression of how my eye would look versus, how I would perform
Yeah.
physically?
Yeah. So there are a number of steps that go on in this. First question is, if I have your DNA, how do I know how is it organized in the cell, right? Which chromosome is it on? Um, so before the Human Genome Project started, uh, researchers used to study genes and chromosomes in isolation. So some researcher will only focus on chromosome X. Some researcher will only focus on chromosome 13. Somebody will be like, okay, I only want to study the eye and I want to understand everything about the eye. So what they would do is they would take some cells from the eye or some particular tissue of the eye, and using biochemical methods, so no sequencing, but they'll use some enzymes to break down those cells, break down the DNA. They will have slow laborious processes called electrophoresis. So it used to be like a agar jelly gel. Do you guys know what an agar gel is? It's like this thick jelly like substance. It looks like aloe vera gel, to be honest. If you like solidified and packed aloe vera gel into a cylinder, that's how yeah, into like a rectangle or a cylindrical shape, that's how it would look like. And what they used to do is they used to treat those cells with particular enzymes that they know cut it into small pieces, run it on the gel, and extract those sequences, and then try and identify, okay, what's going on. Maybe they used to put it in the mice and observe what happened. So maybe with the FLT3, somebody extracted DNA from chromosome 3, put it into the mice and see, there's so many white blood cells now growing in the mice. So it was all trial and error. Like nobody really knew what FLT3 is, but somebody was doing an experiment on white blood cells. Maybe they found out that this particular part of the DNA is leading to more white blood cells, and they named it FLT3. How they named it is a completely different conversation, but all of these genes have been sort of discoveries by scientists doing individual experiments that has all come together into this body of knowledge. You may also ask that when we took blood from these volunteers and just read out the sequence, let's say ATCG, blah blah blah, how do we know that what is the right order that we're reading this in, right? I just know it's ATCG and something something something, but it's like giving me a book, but the pages don't have page numbers. All pages are separate and I don't know which page comes first, which page comes second. So to kind of overcome that issue, what they did is they sequenced overlapping regions of the DNA. So it's like saying, I'll give you that page of the book, but I'll give it to you 4 times. And the first time I'll give you the 1/3, then I'll give you some like overlapping page, then I'll give you another overlapping page, and now you fit it like a jigsaw puzzle. So actually, at the time, researchers also came up with computer algorithms which could find those pieces of jigsaw that fit together and build our genome.